I'm excited to talk to you today about the cerebellum, its involvement in cognitive function, its connections with the neocortex, and importantly, why we should care about its role in development. So I spend a lot of my time thinking about the little brain that's missing from this picture. And the cerebellum, as many of you may know, is a powerful computational structure comprising 80% of the total number of neurons in the human brain, making up a fifth of total brain metabolism. And when flattened, it's estimated at 80% of the surface area of the entire human neocortex. And for about 2 centuries, the scientific community believed the cerebellum was dedicated solely to the control of movement, starting in the 19th century with a French physiologist who discovered that when the cerebella of pigeons were removed, the birds became unbalanced even though they could still move. And then later, a physician on the battlefield of World War I documented a very particular pattern of motor deficits in soldiers who sustained significant injuries to the cerebellum due to Brody helmets that they were wearing, which sat like tin balls on top of the head, exposing the cerebellum to damage from shrapnel. And all of this work really set the dogma that the cerebellum was involved in motor coordination, and for many years, the research community ignored the science that suggested it was involved in other functions. And that was until the 1980s when 3 scientists, Liner, Reiner, and Dow, hypothesized that the cerebellum may contribute to skills that extend beyond motor function into cognitive functions. And they based this idea on anatomical observations, namely that phylogenetically younger parts of the cerebellum, the lateral hemispheres, received a large amount of information from association regions of the cortex, and therefore suggesting that the cerebellum may contribute in some way to these cognitive processes occurring in the cerebral cortex. Indeed, this thought experiment was later corroborated by Strick and others who reported through seminal viral tracing work that the lateral hemispheres of the cerebellum sent projections back to the association cortex, forming a reciprocal closed-loop circuit. And we now know that there are numerous parallel circuits connecting the majority of the neocortex to the cerebellum, which makes these circuits the largest bandwidth of information in the human brain. And a really nice illustration of this connectivity comes from seminal work by Buckner and colleagues who showed using resting state fMRI that the majority of the neocortex is functionally connected to distinct regions of the cerebellum. Now, not only is the cerebellum heavily implicated in cognitive function, but it is also unique in that it has a protracted development, with neurogenesis continuing through the 11th postnatal month, compared with the neocortex, which has largely completed neurogenesis by the 27th prenatal week. And the cerebellum also undergoes an enormous transformation during the 3rd trimester, quadrupling in size and then doubling in size during the first 2 postnatal months. And this increase in volume is underscored by an increase in cell density, myelination, and synaptogenesis. In fact, compared to the pyramidal cell, which has 8,000 synapses, a single Purkinje cell could have approximately 200,000 synapses. Therefore, because of both its protracted development and its enormous connectivity architecture, the cerebellar circuits underlying cognition are likely very vulnerable to dysfunction during sensitive developmental periods. And any perturbation of these circuits likely contributes to the hallmarks of neurodevelopmental disorders like ADHD and autism. And so much of what we know about the cerebellum's involvement in cognitive function has in fact come from fMRI studies. And this line of research all started when Peterson and colleagues published a study back in '89, demonstrating that the cerebellum is activated in response to single word processing, distinctly non-motor tasks. And since this landmark study, many hundreds of imaging papers have been published demonstrating cerebellar activation in response to attention, response reassignment, higher order rule processing, verbal working memory, language, social cognition, amongst many, many more. Now, in a seminal study by Catherine Stooley and Jeremy Schmaman, the same set of participants were tested on 5 unique tasks. Each cluster of colors here represents a unique task, sensory motor, language, spatial, working memory. They interpreted functional activation as being localized to particular cerebellar lobules. So for example, working memory in the violet cluster activates lobule 6 in CRUISE-1. Now, lobules are the typical nomenclature for interpreting cerebellar activation. And there are 10 lobules, starting with lobules 1 to 4 in the anterior lobe, extending through CRUISE-1 and CRUISE-2, all the way to lobule 10 in the posterior lobe. And another way of viewing these lobules is on the flat map representation of the human cerebellum, which allows for results to be shown in one concise view. So our goal, and in the first project that I'll discuss, was to develop a comprehensive parcellation of cerebellar function using a large multi-domain task data set, so extending the work that's been started by Kathleen and Jeremy. So to do this, we took a different approach from conventional methods, and instead we threw the kitchen sink of tasks at the cerebellum in the same set of participants. So we tested multiple domains, from executive function to language perception. Within each domain, we had a range of 2 to 5 tasks, and many of the tasks could be then split further into conditions. So for example, the action observation task was split into dynamic and static conditions. So overall, we had roughly 7 domains, 26 tasks, 47 task conditions, and 24 healthy participants were scanned for 5.5 hours on this battery of tasks, all undergraduate college students. So we thought that this set of tasks would be sufficient to sample the space as richly as possible. So now, to derive a functional parcellation, we first needed to activate the cerebellum as widely as possible. So what I'm showing here are the unthresholded task maps for the group. Warmer colors indicate high levels of activation, cooler colors indicate low levels of activation. And each task condition is contrasted to the mean of all other task conditions. So for the most part, these task maps corroborate what the past 2 decades of neuroimaging results have found. So for example, a verb generation task elicits strong stereotyped lateralized CRUS1, CRUS2 activation, while a working memory task activates CRUS1, CRUS2 bilaterally, and these are well-known patterns of activation. So after demonstrating the heterogeneity of functional activity patterns, we were then interested in leveraging all of these activity patterns to obtain one concise and comprehensive picture of functional organization in the cerebellum. And we did this by decomposing patterns into a region by voxel matrix. We then used a winner-take-all approach to assign each voxel to the region with the highest weight, resulting in this multi-domain task parcellation. Now, one of the advantages of using task-evoked activity patterns is that we can now meaningfully interpret these functional regions using cognitive labels derived from the many tasks we employed. So for example, we can break the verb generation down in terms of the following constructs: word comprehension, verbal fluency, noun reading, visual letter recognition. And we can do this for every task, and then we can learn the mapping between tasks and constructs for each cerebellar region, which allows us to then determine which constructs are most strongly encoded in each functional region of the parcellation. So in applying this method then, what did we learn about feature encoding in the cerebellum? So first we found what you might expect to find, which is that the 2 regions in the anterior lobe, these 2 green regions, encode left and right hand finger presses, and this is a known somatotopy in the cerebellum. Then a 3rd motor region was identified that encoded saccades and other features related to visual recognition. This is the only region of the cerebellum that's known to connect reciprocally to the visual cortex, and this finding is consistent with neurophysiological data from non-human primates, showing this region is associated with ocular motor control. A 4th motor region, an associative motor region, was driven predominantly by action observation, a set of regions that has not really been well described in previous work. But interestingly, beyond these 4 motor regions, the features most descriptive of cerebellar functional organization captured a range of cognitive processes from divided attention to narrative, verbal fluency to autobiographical recall. And we also computed the functional parcellation for individual participants, as although it's largely a practical decision to go to the group, we of course lose information about individual subject level by doing this. And it's worth noting that each parcellation was collected on almost 3 hours worth of data per participant, an amount of scanning time that's usually not feasible or practical for an individual subject. Therefore, there's still utility in a group normative parcellation. So what this map shows at both the group and individual level is that there is a cognitive cerebellum that's likely as important, if not possibly more important, than the motor cerebellum. Of course, missing from this map is another important structure, the neocortex. And we know from both anatomical tracing work and recent functional neuroimaging work that a majority of the neocortex connects reciprocally to distinct regions of the cerebellum. And one typical approach that's been used to map corticocerebellar connectivity is to apply a winner-take-all approach in which each cerebellar voxel is assigned to the cortical network with which it's most correlated. And this is work done by Buckner. However, one challenge with adopting this method is that a one-to-one mapping is imposed between the cortex and the cerebellum, thereby neglecting the possibility that there are alternative mappings between these two structures. So therefore, to go beyond descriptive measures of corticocerebellar connectivity and to quantify this relationship, we needed to build a predictive model. So to do this, we trained a model on cortical regions, averaging the signal within each region, and then we learned the mapping between cortical activity patterns and cerebellar activity patterns. And to capture a range of possible mappings between the cortex and the cerebellum, we trained models at varied sparsity from few to many cortical regions. So the sparsest model was a winner-take-all model, which only allowed for one winning cortical region to estimate cerebellar activity. Then we relaxed the model using regularization to allow for more regions to predict cerebellar activity. We then evaluated the generalizability of these models to new cerebellar data by taking the dot product between new cortical regions and the weights estimated on the training data. So what did we find then? So plotted on the y-axis is the predictive accuracy, which tells us how well our models predicted new cerebellar data. And on the x-axis are the model features, so the number of cortical regions that we used in our models. We also normalized the predictive accuracy by the noise ceiling of both the cortical and cerebellar data. And what we found was that the models that considered a wider range of cortical inputs succeeded in predicting about half of the systematic variance in the cerebellar data compared to the winner-take-all model, which performed worst of all. So all in all, this suggests that a one-to-one model is suboptimal in characterizing corticocerebellar connectivity. So then as a next step, we wanted to visualize the cortical regions functionally connected to distinct functional regions of the cerebellum. So where were inputs to the cerebellum coming from the cortex, in other words? So there's a known somatotopy in the cerebellum, as I previously mentioned, for hand and foot movements. It's noted here by the different colors. And given this somatotopy, as well as the crossed connectivity between M1 and lobules 4 and 5, it was not surprising that the input to the hand regions of the cerebellum, regions 1 and 2, were centered around the contralateral primary sensorimotor cortex, with some additional input from premotor and parietal cortex. We then found that region 3, another motor region located in oculomotor vermis, was predicted by a bilateral set of cortical regions, including frontal eye fields, regions in intraparietal sulcus, and extrastriate visual regions. Region 4, an area strongly activated during action observation, is predicted by a bilateral network of regions, including premotor cortex, supplementary motor cortex, and parietal cortex. And beyond these motor regions, we then wanted to know whether the pattern of connectivity differed for functional regions in CRUISE 1 and CRUISE 2, the traditional cognitive regions of the cerebellum. And overall, what we found is that these cognitive regions received convergent information from many areas of the frontal and parietal networks. Again, driving home the point, the cerebellum receives a large amount of information from all of association cortex. So then, to summarize, I set out to investigate corticocerebellar contributions to cognition. I showed that the majority of regions in the human cerebellum are activated by non-motor or cognitive processes. And the highest convergence of information from the neocortex is then observed in regions of the cerebellum that are strongly associated with cognitive processes. Finally, a quantitative model of corticocerebellar connectivity can explain about 50% of systematic variation in the cerebellum, suggesting that the cerebellum is really doing something that cannot be explained by cortical processing. So there are unique computational functions being performed by the cerebellum. So all in all, I hope that some of the work that I've presented offers an insight into the possibility and the promise that the human cerebellum can offer in understanding the developmental processes that underlie cognitive function. And perhaps it provides a foundation for some of the work that my colleagues will be presenting during this symposium. And with that, I would like to acknowledge my PhD advisor, Richard Ivory, collaborators from Western University, Stanford University, and research assistants whose contribution to these projects were really, really valuable. And thank you for your time.